if it looks like we're behind after a few more lectures, then I'll have another online lecture for this class on Monday. We'll play that by ear for a few days here.
Yeah, me.
The last time we were together, we talked about the process of transmitting sound waves into the inner ear, and we finished that conversation with bending hair cells, if you remember, that are sticking into the tectorial membrane of the orbital cortege. That's really where hearing happens, where there's a conversion of this mechanical process by which sound waves cause the movement of these structures, including the tympanic membrane, the three bones, the ear cavity, and then the vibrations of stapes in the oval window creating movement in the fluid inside the inner ear, physically pressing on the tectorial membrane of the orbital cortege, and then that's what converts. It's that movement of those cilia or hair cells in the tectorial membrane that actually changes the conductance. It causes action potentials in the auditory or the vestibulocochlear nerve. So that's the process we did last time. The reason this picture is back up again today is because I want to talk about balance. Same sort of thing, do the physiology of balance, and really we're going in the same direction here. The phenomenon is very similar, just some different terminology. The phenomenon is basically going to be a mechanical process by which you move fluid inside these structures, and the movement of that fluid, or the weight of the membrane, really of the first set, actually bends hair cells. And so a little different terminology there, the kinocilium and the sterocilium, but essentially we're talking about the same physiology. The bending of those things changes the conductance in the nerve cell. It's really a modified epithelial cell is what it looks like, but then you get sodium entry into the cell, you kick off action potential, and that is interpreted by our brain as balance. So very similar phenomenon here, bending these hair cells, changing the conductance. That's where the physiology really is converting the mechanical stuff that's going on, the movement of the fluid, the vibrations, etc., into the physiology of action potentials, because that's how we hear, that's how we have balance, is the transmission of action potentials along this vestibulocochlear nerve. All right, now the first one is a structure here in the middle portion of the inner ear called the vestibule, hence the name vestibulocochlear on the nerve. So this middle portion here has two distinct structures in it, one called the utricle and the other one called the saccule, and they are positioned essentially at right angles within this middle portion of the inner ear, the utricle and the saccule. Each of them has hair cells, as I just indicated to you, extending off of what looks to be modified epithelial cells. This is an electron photomicrograph of it, but this shows you the real physiology here. It's actually the bending of those things that changes the number of action potentials that are occurring in the nerve. And so you can see that illustrated here. Whenever the smaller cells, they're typically referred to as the stereocilia, are bent toward the larger one, the bulb on the end of it, called the kinocilium. Whenever that happens, you get an increase in the number of action potentials. When it bends the other way, you inhibit it, you get a decrease in the number of action potentials. Y'all, this is how we do balance. That's what balance is right there. It's the number of action potentials in the nerve caused by the bending of these specialized cells within the utricle and the saccule. Now let's talk about that vestibule apparatus a little bit more. So you see the basic physiology here is simply a mechanical process, bending these things. And so we know that the utricle and the saccule, located in the vestibule, are angled against one another at almost right angles. It's almost a 90-degree angle. This one shows exactly, but this is really intended to be an illustration for one of them. The illustration here is to show you the phenomenon that's occurring here in that portion of the inner ear. The phenomenon is this. Instead of it being dependent on the movement of fluid here, these structures are actually telling you where your head is. Whether it's upright, bent forward, bent sideways. The way that happens is the membrane, we called it the tectorial membrane in the orbitocorti. Now in the utriculosaccule we call them otolithic membranes, because otolith means rock, and you can see these guys have rocks, calcium carbonate crystals in the membrane that give them weight. And so you can see that if the head is upright, this particular organ, this one would be the ventricle, it doesn't matter. This one is sitting horizontal. The otolithic membrane is not causing the bending of the kinocilium or the stereocilium. But when the head is bent forward, you can see the weight of this membrane now causes the bending of those cells. So this is the mechanical process here. The otolithic membrane bends these guys, and when you do, this is what happens. If you bend toward the kinocilium, more action potentials. If you bend away from it, fewer action potentials. And this is the fundamental physiology for how we know about balance. Your textbook description is a good one, about balance in what we think of as static equilibrium or linear movement. So my head is upright, static, I'm still, but I know where my head is. My head is bent forward, I know where my head is. If I move linearly, in a straight line, these things do the same thing. It's the movement of the weight on those membranes that conveys that information into our brains.
So is there certain different pathways to balance, or does sight have something to do with this? Because I mean, if I close my eyes, I mean, I can try to balance, but I mean, it's still...
Yeah, it's a very good question, Dakota, and I'm going to address it here in just a moment, because that's really common sense for most people. They think, well, balance is with vision. People also know if I have an inner ear problem sometimes, I can have balance problems. So generally people know that there's some kind of combination going on here. I think we can do a little better than that in a physiology course. So just give me just a moment, and I'm going to show you those pathways. You are absolutely right. There's a mixture of information here between your visual system and the locations of the pathways for these vestibulocochlear fibers. I did teach them to you in the fall, the pathways at least, where the cochlear nucleus is in the medulla oblongata, and then how that cochlear nucleus presents its information upstream. I also showed you the vestibular nucleus, which is just slightly medial to that, and how that information is transmitted upstream. So we're going to talk about it again. I will not force you to learn the specifics once again of the names of the waypoints, but you at least need to think about it exactly as the way you articulate it. Vision is playing a role here. Okay, good. So now here's the other structure. This is the base of each of those semicircular canals. And so once again, let's return to the first picture. Here are the semicircular canals, and they are oriented in the three body planes. And at the base of each of these semicircular canals, you can see there's a swollen piece of this. This swollen piece is called an ampulla. With each of these structures, I should mention again the anatomy of this. We saw a soft duct within the hard bony structure here. The utricle and saccule are essentially soft ducts within the hard bony structure here, and there is a soft duct within these semicircular canals also. And it's enclosed. There's fluid inside it, there's fluid outside it. And we learned those in the fall. We call the scala vestibuli, we said has parallel. The scala tympani has parallel. We said the cochlear duct has endolith. It's a slightly different consistency of fluid. But I wanted to remind you here, there's soft ducts inside these hard bony structures. And so this is a picture actually of the end of that soft duct to talk about the physiology, where there's a swollen piece called the ampulla. Now the terminology is a little different here. Whereas the tectorial membrane was the structure that helps bend the cilia in the orbitocorti for hearing, the otolithic membrane bends the stereocilia and kinocilia for static equilibrium. Here, the membrane here is called the cupula. This larger structure here, where all this is happening, is known as the crista ampullaris, but the membrane that the fluid is going to move is called the cupula. That word means sail. And so the idea here is as the fluid moves through the semicircular canals, depending on the rotation of your head, this is not linear movement now, this is rotational information. The fluid in each of these semicircular canals, oriented in the three body planes, will rotate in those canals based on the rotation of your head. And when it does so, it bends the cupula, catches the fluid like a sail, and it bends these hair cells of these modified epithelial cells and sends information to the brain. So this is a blow up out of here. You can see the cupula here and how they would bend whenever the fluid passes through this membrane. It catches the hair cells and bends them. Now this is a question that Dakota asked, and he is absolutely spot on. The key moment in balance is a group of nuclei found in the brainstem. Now, a good anatomy student who had a decent anatomy teacher will be able to say something more here. When I say the vestibular nuclei of the brainstem, I'm talking about the nuclei that you learned in the fall that receive information from cranial nerve 8, the vestibulocochlear nerve. But that's really where we've been putting all these action potentials. Now, where in the brainstem are these nuclei? Can you help me remember that? Where are these vestibular nuclei? The brainstem is made of, it was the green parts of the structure of the picture we looked at. The brainstem is midbrain, pons, and medulla. This is what we mean. You say you have, this individual has a tumor in their brainstem, okay? Anybody who's had anatomy should know immediately why no surgeon wants to mess with a tumor in this area. So I taught you a whole bunch of really important things that were in the brainstem. This was one of them, the vestibular nuclei. Where in the brainstem? They're in very close proximity to the cochlear nuclei, but they're not the same. There's a division here between the hearing information and the balance information. These were in the medulla oblongata of the brainstem. Does that ring a bell for you? We looked at coronal sections so that I could teach you those in the fall. Okay, but Dakota's question was, what about vision? And he's absolutely right. You can see that the vestibular nuclei of the brainstem also receive information from your eyes. Now, you also should be able to track this pathway backwards from the eyes. In fact, I've taken you back as far as the occipital cortex in this class with visual information. So that shouldn't really be a surprise to you. The vestibular nuclei receive information from the eyes. You also have learned, as we just pointed out, the vestibular apparatus feeds those. So this was the primary pathway here. You also know that the cerebellum is involved in controlling muscle contraction, right? You know that it has to receive information from proprioceptors to help you with balance, locating in space. And so the vestibular nuclei is combining information from the vestibule and the semicircular canals and the eyes and the cerebellum and from joint, tendon, muscle, and cutaneous receptors in the body. There's a lot of information here in the brainstem about balance. And then we know from these vestibular nuclei, there are motor pieces of information now that are going to go to the important places that do balance for us. This is the functional pathways here. This is the sensory stuff feeding the vestibular nuclei. And then down here, we're going to take this information to the oculomotor center. The oculomotor center. Where is that? Okay, good. So cranial nerve 3 contains oculomotor information. That's true. That's the oculomotor nerve. But where in the brain is all this? Okay, there is some information here in the superior colliculus that does some of this, actually. That's true. That's part of it. Good. So just posterior to the premotor cortex, there's a frontal eye field that's also connected to the superior colliculus. So this is a one arrow thing, but there's a lot going on here. the ocular center of the brain, including all these various structures that control the eyes. So really, if I think about the vestibular nuclei influencing the movement of my eyes, then I have to get to places in the brain that feed information to 3 different cranial nerves. And you named one of them, cranial nerve 3. What are the other 2? What did you say? Okay, but I need names if you don't mind. He said the name for cranial nerve 3 is the oculomotor. 4 is the trochlear. 6 is the abducens. Okay, so the oculomotor center is going to use those 3 cranial nerves to move your eyes. And this is something that I want to ask you to demonstrate in the lab tomorrow. Not only with the reading stuff that you're going to do, but I'd also like for you to be able to demonstrate to each other a vestibular nystagmus. Nystagmus, a nystagmus, which is an unusual jerking motion of the eyes caused by a balance problem. Now, I have historically been very hesitant to do this in the laboratory. I had a kid get hurt one time in this hallway doing this. But I'm going to bring a stool in tomorrow, and I'm going to supervise it because I'd like you to be able to see it. If I teach the pathway in the lecture portion and then you see it happen in the lab, then it will leave an impression on you. So basically what we'll do is we'll put someone, probably one of the boys, we'll have a volunteer, and we'll put you on a stool, and then several of us will get around you. And then we will turn you, close your eyes, and we will turn you until it's, we've made you sufficiently dizzy. The lab procedure asks you to turn the student until the sensation of turning stops. Yes? Oh, yeah. Now, could you explain to someone why the sensation of turning would stop?
Because you get so used to turning that your body acclimates itself to it.
Okay, but I think you've learned something today that can help with that. So which portion of your inner ear is doing that? The semicircular canals. Good. Okay. What is the name of the membrane? So when I turn this way, the fluid goes that way, right? And it hits the cupula and bends those cells, and it tells you, that's the way your brain knows I'm turning this way. But after you turn that way for what?
The fluid will stay.
Yeah, the fluid that had this reflexive backwards motion will catch up with your turning. And the hair cells will then straighten back up, right? Now, when you stop the turning, what happens?
Then they float again.
They bend over the other way, and that tells your brain that you're off balance. Yes, you're standing straight up, but it wants to push you the other way. Right, so it becomes very difficult to keep your balance. Now, that is true, and we're going to hold you, whoever does this. I'm not going to require you to do it, but you volunteered. We're going to hold you so that you don't fall, okay? And then we're going to ask you to open your eyes. When you open your eyes, we're all going to be staring at you. Because your eyes are going to be doing a nystagmus, jerking like this. They jerk in the motion of the rotation fast and then a slow return. Jerk, 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 like that. A vestibular nystagmus. And this is the reason why. Because that information for balance is being fed to the ocular center to control the eyes. Okay, now, we also know that this information is going to the spinal cord. Now, we just said it, but what would be the reason for this information? Yeah, you're going to have to send information to skeletal muscles to do the balance, right? Okay, so this is a broad view of neural pathways for balance information. It's a very good one because it talks about all the main pieces that are used by the body to do balance. All right, the last couple special senses. This one is for taste. I think Dr. Du Bois spends some time with students. I filled in a little bit for her a year or two ago when we were doing taste buds. Taste information is interesting because most people think we have a lot of different taste buds because we have so many different things that we think we can taste. The truth is there are only arguably 4 or 5. Most textbooks will have 5 now because of that Japanese group that identified the umami receptor. But at the very least, there are 4 that have been known for a long time, and they are sweet, sour, bitter, and salt. Sweet, sour, bitter, and salt. These are the 4 standard taste buds. Now, in order to, okay, so let's say the other piece of that before I go and show you how physiology works. The taste that most people think is taste is actually smell. So all of the varieties of flavors that we are able to detect is actually olfaction, because the taste buds just do not, they can't, you cannot get taste buds to give that kind of information to the brain. There aren't enough different varieties of them. And we know the physiology behind these taste buds, and here are the big 4, salt, sour, sweet, and bitter. We know that the salt ones make perfect sense, so if you break these things down molecularly, you can actually identify the proteins that are in the membranes. And so the question is, right, for every single one of these, the question has been, how does the sensation, the special sense, get converted into actual flavors? So that's how we finish this. In order to get the salt information to your brain, it is the entry of sodium. That makes perfect sense. That is how you make the action potential. In order to make the sour information go to your brain, those are hydrogen ions, acids, right? Increases in hydrogen ion content. And the hydrogen ions, there are proteins that allow the hydrogens. Have you seen a protein that will transport hydrogen ions? There's no, I do not do nervous and kidney first randomly in this class. Once you have learned about all these proteins that allow these things to move, it just never goes away for the rest of physiology. So absolutely, hydrogen ions can cause action potentials. You know that already. Sweet is a G protein mechanism that uses cyclic AMP and closes a potassium channel. Why would closing a potassium channel cause an action potential? Have you seen that before? This is why I told you what I did when we were going through this. If you learn the sodium, potassium, calcium, chloride story, then you can explain anything. Okay, and then the bitter uses the G protein mechanism for a different second messenger system. Now, notice I've mentioned cyclic AMP, IP3, and DAG here as second messengers, and I've only briefly alluded to them. I briefly alluded to them when we talked about adrenergic binding. And I said there's a whole bunch of different ways you can get to the channels using the second messengers. So once again, I'm going to briefly allude to them. It's not time yet to learn all the details. We're gonna keep poking at you with these second messenger systems. And then when I get to the endocrine system, I'm going to walk through these 2. But here they are. This is the cyclic AMP pathway. And here are all my hormones that I want to talk about later. And you can see the hormone binding to the receptor. We looked at this picture earlier in this class, and I said, that's norepinephrine binding to a beta-1 adrenergic receptor. Yes? Now what am I saying? This is the sweet molecules dissolved in the liquid, binding to their receptors in the taste bud, or the bitter molecules. This is the base stuff, binding to the dissolved in the liquid, binding to the receptor, initiating a G protein mechanism, and then ultimately, cyclic AMP is going to activate a kinase. Now, I made a big deal about this also. The kinases are for the purpose of phosphorylation. And where this lists enzymes and cellular secretions and ion channels, I'm going to want you in the endocrine system to be able to talk about this some more. For example, when we did norepinephrine before, I took you through and said, this is going to increase heart rate. Okay. Well, But when we do this the next time, I'm going to do muscle next, and then endocrines right after that. So when we do this the next time, I'm going to identify these things for you. I'm going to say, what did you phosphorylate? And what did that cause? Okay, so once again, you've seen it with your eyes. This is the cyclic AMP pathway actually closes the potassium channel. So that would be this effect right here on ion channels. Okay, and here's the DAG pathway right here. Once again, a G protein is involved with this. Different enzymes this time, phospholipase instead of adenylate cyclase, but you can see that there are second messengers that are going to act on kinases here. So it's a little bit different in the way this is done, but we will want to walk through these sequentially so that you guys can speak intelligently about at least these two second messenger systems. So I'd like to introduce them again here for sweet and bitter for taste. Now, the last thing here for taste is something you learned in the fall also, and that is that taste information from the taste buds actually reaches the brain. It actually reaches the brain. This should look, when I look up at this, I go, these kids should be going, I've seen all those words. Facial, glossopharyngeal, and vagus. Yeah? Front to back, 7, 9, and 10. Does that ring bells for you? Passing through the brainstem. I did not this fall make a big deal about the solitary nucleus. It was on a picture we looked at, but I didn't emphasize it. But you did see this one. The thalamus relays all this information. And for taste information, it ends up in the insula, right? There's a gap here between the parietal and temporal lobes, the lateral sulcus. And if you go inside there, there's cortex buried inside the cerebrum. We call it the insular cortex. And that's where that's where taste information is located. The gustatory cortex, within the cortex of the insula, inside the lateral sulcus. 7, 9, and 10. Let's review. All right. Now, the last one here is smell. Smell is a remarkable physiology. A gentleman by the name of Gordon Shepherd wrote a very narrow text that we used many years ago in graduate school. There is a new edition of that out. I looked at Amazon, and I heard him give a talk one time talking about all the different proteins that are present in the neurons for smell. It is a fantastically complicated process, fantastically elegant, and somewhat unexpected, I would say. The Human Genome Project revealed something in the neighborhood of 45 to 50,000 genes in the human genome. Something like that. 48,000 something is the last number I saw. 1,000 of those, are you getting the sense from me on this? 1,000 of those genes are genes for proteins that are used for smell. So there's nothing close to that in any of the other special senses. It's a fantastically complex pathway. Most textbooks, most freshman textbooks, use these multiple genes for receptors for smell, and there's discussion of gene duplication. I don't know if y'all did much of that with Dr. Criswell in freshman biology, gene duplication stuff. So,
Yeah, we've had origin ones.
oh, good. So you talk about orthologous and paralogous genes.
Not
that.
Talk about gene duplication,
So you talk about exaptation.
where they can change the one gene to do like this and the other one can put a different one on it.
Okay, I don't understand that. Okay.
Yes.
Okay, so did he talk about the type 3 secretory system? No, okay. Okay. I'm trying to
Obviously, we did talk about gene duplication.
Okay, so what did you talk about exactly when you said gene duplication? In what context?
Of mutations and how we can go from one thing to
Okay,
another. And the final answer was really
okay.
as usual.
Okay, so this is a very common argument today in science textbooks for how things change. this gene duplication. And these guys, as you can see, there's 1,000 of them, so they're normally brought up. Did you bring up the olfactory genes? Okay, well, I am completely bombing on that, but at least you've heard about gene duplication.
Heard the word.
Okay, so at the very least, the idea there is, and y'all can help me describe this, Dr. Criswell has actually written about it. The idea here is that if you have genes that are producing proteins, for which there are many different varieties, there is at least a possibility that some of those genes could mutate to make proteins that would do other things without any loss of function. So that you could essentially, because you have multiple copies, you could use these proteins for other functional things, which would give the organism an advantage in a Darwinian sense and allow them to evolve some new functional capacity. Does that sound more familiar? Okay, all right, so I finally hit on it. But as I'm sure Dr. Criswell showed you, there's a major problem with that.
There's too many redundancies built into the system, so it actually.
Exactly. It's very problematic because theoretically it sounds very nice, but in practical terms it can't be done. And did he mention Doug Axe's series of papers in Cell? Okay, so Dr. Axe, who was here in the fall, if you want to meet him, actually published a series of papers in Cell Biology where he actually tested the possibility of evolving in two very similar proteins in a gene duplication experiment, evolving the ability of one protein having an enzymatic active site into another protein with a different enzymatic active site that are essentially identical to each other but functionally doing different things. And it could not be done in the lab. He published this work, and the evolutionary biology community completely ignored it. The implications there are that living systems with gene duplication things do not have the capacity to evolve new functional proteins with new enzymatic active sites. So it's a problem today that you will not learn about at any other school except Louisiana College. Nobody wants to say this anymore. Incidentally, if you would like to watch, I think about this when I'm in the public. I do not want to embarrass myself, and I do not want to embarrass my students, and I do not want to embarrass my college. Monday, I confronted, before a live streaming camera, I confronted the evolutionary biologists writing the standards for the state of Louisiana. If you go to Twitter, the Louisiana Department of Education, you can watch that if you'd like to see it. I asked two questions in sequence of these folks about language in the standards concerning evolutionary biology. And the only response I got for the first question, I had 7 of them prepared to ask them. The only response I got to the first question, which was about 7 appearances in the fossil record, was we contacted the leaders of the National Academy of Sciences and other major scientific organizations, and they were uncomfortable with the language that you had put in. I put 2 words. All this list about the fossils, extinction, change of life forms, all these different, the whole list, and I added 7 appearances. I would like for the young people in Louisiana to know about the Cambrian explosion, that it is evidence that most of the animal phyla all appear at one time in the fossil record. Why can they not know this? Everybody knows it in the scientific community. I provided a dozen different papers with it published. Why can they not know this? Lady from LSUA, Brenda Nixon, who's giving a plenary lecture out there next month for evolutionary biology, says to me out loud, on camera now, most scientists are not convinced that 7 appearances is actually an observation in the fossil record. And I said, whoa, whoa, whoa. I said, did you read the scientific literature that I provided on this matter? And she immediately started crawfishing. I didn't mean to say, I didn't think that you meant to say that out loud. I think that you misspoke that, right? Yes, I misspoke that. The second question I asked was, I would like to have a standard, the standard put in for building phylogenetic trees. The standard said that students were to use different techniques, morphology, molecular biology, to construct phylogenetic trees to demonstrate evolutionary relationship. I provided, again, dozens of papers where different techniques used to demonstrate phylogenetic comparisons disagree with one another. In fact, dramatically, there are some techniques that are used that would suggest that we are, as human beings, as closely evolutionarily related to blue-green algae as we are to primates. No moron would suggest that that is true. So I wanted language put in there, just 4 words, that there is just a wording that would allow young people to know that there is conflict between the demonstration of phylogenesis from different techniques. The head of the biology department at A was there, and she says, I'm a molecular biologist by training, and I'm uncomfortable with that language. And I said, when I brought it up, I said, Dr. Sullivan, I would love for you to instruct me on why this language cannot be put in. Could you make a comment about that? And she said, out loud, she said, I am more than willing to give you instruction in private. Here's your opportunity. I have asked this question out loud in public. The cameras are rolling. If I need to be corrected, now is the time. Instruct me. Am I the stupid one here? Silence. Not a word. The director of the whole thing says, I have no explanation for that. I'm the only dissenting voice in the room. There's 60 people. I don't get it, people. I do not get it, young people. I do not get it. Makes me crazy.
So who makes the final say on what's actually put
The
out?
say has already been said.
So it just doesn't get put out.
I was outvoted. It's a done deal. Yes, very disturbing.
So all the efforts this past semester and stuff are pretty much for nothing?
At this point, for nothing.
But when you mean at this point, does it mean that you're done with it, or is there anything else that you can do?
Well, I think there are some politicians that are going to try to raise a stink whenever the standards come before the Board of Elementary and Secondary Education. But I think it'll go from all at this point. The only positive I can take away from it is there were a number of people on the committee who came to me afterwards. So the Baton Rouge Advocate interviewed me and quoted me yesterday in the paper. And they did a fair job. And of course, the Baptist Message is going to write an article on it. And then there were several people in the room who were science teachers, 8th grade high school science teachers, and they said, could you provide some of this information for us? So all I can hope for is that there will be some high schools who will be willing to give a fair presentation of the science. I was not asking these people not to teach evolutionary biology. That's not the point. I would just like for them to also reveal to these young people that there are hundreds and hundreds of scientists and hundreds of peer-reviewed literature articles that are in conflict with the current Darwinian model. Why can't they know that? Why must we hide this from young people? It's stuff that keeps me up at night. It makes acid levels rise in my gut when I think about it. Y'all got me off track. So 1,000 genes per receptors, 10,000 different odors detectable. This is the pathway that I taught you in the fall. Bipolar neurons there, yeah? In the olfactory system. They are the receptors. Things have to dissolve in a liquid, and then they activate these various protein receptors. All right.
So when you get hit in the face with a softball and it messes up your sense of smell, like you can't taste things right, what part of that is like injury? Like, you know what I mean? What causes that?
Yeah, I think, well, it has to be something in this, why is this not working? It sounded like it was experience.
Oh, why does your smell affect your taste so much? Like, I got the sinus surgery, so they cut out bone and took out a bunch of stuff, and I had to get a tire replaced. I went into this tire shop, and I kid you not, I could taste the rubber.
Oh, yeah.
How does
Yeah,
that work?
so you're just saying the words taste and smell because you think you're tasting it. You're smelling it, right? And so anything that would damage this, right, Hannah? Anything that would damage this pathway is going to have impact.
So it's not one specific region, it's just something in there got messed up.
Yeah. Absolutely, that's a good way to say it, something that they got messed up. Now, this pathway is also should look very familiar. This is the basics of the smell. Here's the odor chemical binding to the protein, and you can see there's my second messenger system, my cyclohexane system, and then where do we finish with this? The sodium channel. Yeah, so this is what I meant when I was telling you guys before about the influence of sodium, potassium, calcium, and chloride. We just keep seeing these things as we go through different pieces of physiology. All right, the next content is muscle. So this will be next week's lab. Next Thursday, we'll do a lab on muscle. And this is, you guys learned a lot of muscles in the fall. You learned, Abby sent me, Abby Mallet, you know, she's in PT school now, she sent me her first anatomy lab exam. Took a picture of it because she made like second highest grade in class, and her first gross anatomy test. And she said she wanted me to see all the names on it. She said, "Look at this exam. There was nothing new here." And she wrote just said thanks because she had a good anatomy teacher. And I told her that she made my day. Incidentally, I told her, and you guys can know it as well, this summer I normally teach the nursing A&P during the summer, but St. Augustine has contacted me now because we've had several LC kids go through their program. There's a kid, Nikki Bowen, who took the sophomore class, non-traditional student, so she's doing an online physical therapy degree. Y'all heard of this? Emily Bro did it, and now Nikki Bowen's doing it through St. Augustine through Austin, Texas. And the director there, Tom Werner, called me a couple months ago, and he said, I've seen enough of the Louisiana College students and their anatomy abilities. Would you be willing to teach gross to the PT kids this summer? And I said, as a matter of fact, I would. I would love to do that. So I think we'll still be able to teach AMP here. We'll get an adjunct thing to teach it. But if any of you guys end up in PT school, it's possible that I can teach you anatomy at an undergraduate level and as doctoral students coming up in the years to come. So that sounded like sarcasm.
Very much so.
I read that correctly? Okay. Well, that was supposed to make you think, oh boy, I didn't work that way. Fair enough. Okay. All right. So anyway, I don't know what I got on that. I've had a bad day here. I'm having group therapy. I don't know if y'all know. Hey, I've been so frustrated over the past, day or so, and so down. My girl came up to console me yesterday. She knew I got my behind handed to me on Monday, and so I've used you young people this morning to provide therapy for me, and I appreciate that.
Now I have to go to LSUA and take a class and actually look these teachers in the face. And
You have to now?
yeah, I have it next. I have to go there for a class now.
Right.
This is different. I have to see these people that you just
Call out.
then I gotta think, man,
Yeah.
you're why all these problems.
Yeah, well. I'm not saying they're not good educators.
Yeah.
I'm just saying we have some disagreements. That's all. Okay. So let's start talking about the physiology of muscle. I always like to introduce muscle physiology with the concept of motor units. This is a really good way to think about how you control muscles. What you've learned from me so far is many of the pathways that go to skeletal muscles that make them contract, and you've learned the connections, the origins and insertions of those muscles. You've learned the innervations and the connections. But now you need to learn about what makes them work. So this is a great picture to do that with, I think, to talk about motor unit phenomena. As it turns out, the neurons that are in the ventral horn of the spinal cord, neurons that we call lower motor neurons, the LMNs of the ventral horn, as it turns out, these neurons are the ones that send the axons to the muscles. That was a major part of the pathway. In fact, most of them synapse here for the first time, which means these are the second order neurons. Where are the cell bodies of the primary neurons? No. If this is the second order neuron right here, where's the first one? Okay, there is crossing there, but I'm asking you, where's the cell body? That's the axons cross in the pyramids. That's true.
Pyramidal.
They're pyramidal neurons located in the cerebral cortex, specifically the precentral gyrus. Yeah, and so these are the second order neurons here that are then sending information. So that picture right there is intended to be used to teach students reflex pathways. So this is a Marriott picture right here out of the IP modules. And so you can actually teach kids reflex pathways using this guy right here. This would be a polysynaptic. Yeah, PhysioX is not the modules. PhysioX is experiments, but not the module sets are. It's the other one, yeah. So anyway, these are the second order neurons in the ventral horn, and you can see they're projecting out to muscle. Now notice there's more than one motor neuron here. That's an important physiological observation. More than one motor neuron to a muscle. We say by definition that a motor unit is one motor neuron and all of the muscle cells that it contacts. And the way the physiology works for muscles is really tied up in this concept. So I can put these things in categories. There are 4 different major groupings of muscle cell types that are controlled in different ways. But I think I can build a platform from which you can use to learn any of that later. This is the platform I like to use. These things go together, all right? We know that most of the time in larger muscles, the vastus medialis, the rectus femoris, the sartorius, right? These large muscles, we know that most of the time, motor units are huge. A single motor neuron coming from a single cell body in the ventral horn will contact a lot of muscle cells, which means if the action potential gets generated in that guy, all of these muscle cells are going to contract. This is gross control. Huge motor units. For large muscles, proportionately, there are fewer numbers of motor neurons controlling those. So you don't need fine control of a vastus medialis. You want the whole muscle to contract. So there are proportionately fewer numbers of motor neurons, larger motor units. Now those muscles that are larger in your body, generally speaking, look different than the smaller ones. They have a deeper, darker red color to them. And there are different ways to refer to these. I'm going to call them, because every textbook has some version of this. I'm going to call these guys the large muscles with the large motor units, proportionately smaller numbers of motor units and motor neurons. That is, one motor neuron contacts more cells. These muscles tend to have more stored oxygen in them. They tend to have more endurance. And the oxygen is stored on a binding protein that is the cousin, it's the first cousin of the blood binding protein for oxygen, hemoglobin. It's called myoglobin. So, if you see a large muscle, it will normally be a darker red color because it has more myoglobin and it has more oxygen carrying capacity, it has more endurance, it will contract longer, and it has larger motor units. It has fewer motor units per muscle. But the motor units themselves are single motor neurons that contact a lot of muscle cells. If you see in the categories, I want you to think in those categories. Large muscle, red, myoglobin, oxygen storage. We call those muscle fibers slow twitch fibers. Slow twitch. Some texts refer to them as red fibers. Some of them have type A's or type B's on them. I don't want to do that. But red or slow twitch, high myoglobin, high oxygen content, large motor units. That's the control and the appearance of the muscle. On the other hand, smaller muscles, like all the pollicis muscles in your fingers, the extrinsic eye muscles, if you look at them, they have a much lighter appearance. They're almost white sometimes. If you look at the numbers of motor units that control them, you will find that a motor unit there is very small. In other words, a single motor, a single axon coming out of the cord will control a very small number of cells, just a few, maybe 2 or 3, maybe 1 sometimes, where a single motor neuron contacts a single muscle cell, or just a couple. What that does for a smaller muscle is it gives more control. You can isolate control. Now, normally, generally speaking, those with smaller motor smaller motor units, those generally speaking are lighter in color, which means they also have less myoglobin and less oxygen storing capacity and less endurance. So they wear out faster. Small muscles you have more control of because of the motor unit phenomenon, but they have sacrificed control for oxygen storage, and oxygen is energy. So I'm going to teach you that in this section. So this is the way I like to bring it up. Now, the other phenomenon here with motor units is equally as interesting, and that is we actually can control the strength of even a large muscle contraction. In other words, if I want to pick up a lightweight, then I don't need all of the muscle cells. I need a few. And so the system is set up so that some of the motor neurons of the ventral horn are more sensitive than others. You explain that? How would they be more sensitive?
Resting membrane potential.
Yes, you would have a slight difference in resting membrane potential, and we know that's true. What else? Why would they have a difference in resting membrane potential?
They have more sodium, potassium, voltage, ion channels on the axon helix.
Yes, that's exactly right. And so it's a differentiation in the protein content of these neuron cell bodies here. Some of them, they're ready to go. It's easier to get them to the threshold. Those will be the ones that will go first. You'll use those to do a lightweight. But if you want to pick up a heavyweight, then you want to use more, right? So you will stimulate more of these guys. That is a phenomenon in physiology for muscles called the recruitment of motor units. You recruit more of them when you want to make a stronger contraction. Very elegant process. And once you've had neurophys, you're able to explain those. That's great. Okay, so motor neurons and motor units. Now, when we pick this up, I got to shut up. When we pick this up next time, I'm going to go to the muscle. You recognize this. I taught you the differences in how motor neurons connect to skeletal versus how they connect to single unit versus how they connect to multi-unit versus how they connect to cardiac. Remember that? Remember all the varicosity stuff and how the neurotransmitter? All right. This will be the skeletal muscle story. And so we're going to sequence this. It will be very, very useful for you. Maybe after lab tomorrow or Friday morning sometime to watch this on the Mary CD. I'm going to go through slide by slide by slide with her for the next 5 or 6 slides to teach you how this works. We're going to have to release a neurotransmitter. It's going to be acetylcholine. We're going to have to bind it to a nicotinic cholinergic receptor. We're going to have to make an action potential in the cell in the sarcolemma, and we're going to have to spread that action potential down to where the proteins are in the cytoplasm to make the muscle work. So that's what we're going to do on Friday.